Good morning, students. We'll continue the topic chemistry of proteins, competition number 5.2. Here today we are studying definition and classification of proteins with various examples. The word protein is derived from Greek word proteos, which means primary. Primary means very important. As the name shows, the proteins are of paramount importance for biological systems. Definition of protein is very simple. Proteins are polymers of amino acids. Out of total dry weight, 3/4 of our body weight is made up of proteins. Proteins are used for bodybuilding. All major structural and functional aspects of the body are carried out by protein molecules. Amino acid sequence in a protein determines the functions of the proteins. Coming to the classification, proteins are classified in different manners. So there are 4 different ways of classification of proteins. We'll study one by one. First is on the basis of size and shape. On the basis of size and shape, proteins are of two types: fibrous proteins and globular proteins. Fibrous protein means a proteins which has length to width ratio more than 10. They are elongated and needle shaped. They resist digestion by digestive enzymes, possess secondary structure, and examples of this type of proteins are collagen, elastin, and keratin. Globular proteins means the proteins where length to width ratio is less than 10. They are spherical and oval shaped, soluble and easily digested by digesting enzyme, possess generally tertiary structure, example albumin, globulin, and enzymes. Next classification of protein is on the basis of functions of proteins. Proteins performs various functions like catalysis, so they are called as catalytic proteins, example enzymes. Some proteins form structures called as structural proteins, example collagen, elastin. Contractile proteins means which helps in contraction and relaxation in muscles, example actin, myosin. Some proteins transport various substances, they are called as transport proteins, example hemoglobin, albumin. Some proteins stores various substances, they are called as storage proteins, example myoglobin and ferritin. Regulatory proteins means the proteins which regulate the functions of cell, example hormones. Genetic proteins are the proteins which associated with DNA, example histones. And defense proteins which protect our body from antigens, example immunoglobulins. Next classification of protein is on the basis of nutritional value. We classified proteins into 3 categories. Complete proteins first means the proteins which contains all essential amino acids. Examples of these complete proteins are lactalbumin, O-albumin, casein of milk, and egg albumin. If we consume this complete protein, nitrogen content of our body increases and we will gain weight. Next category is incomplete proteins. One of the essential amino acid is deficient. Example, pulses proteins are deficient in methionine and cereal proteins are deficient in lysine. Now if you consume this type of protein alone, then your nitrogen balance of the body does not increases and you will not gain weight. But in this case, if we combine cereals with pulses proteins, then the deficiency of one is fulfilled by the deficiency of other proteins, and hence this is called as mutual supplementation. If we consume both cereals and pulses, then we will gain a weight and body will have positive nitrogen balance. But if you consume alone only cereals or only pulses, then you will not gain a weight. Next class of protein is poor proteins. The proteins which lack two or more essential amino acids, they are called as poor proteins. Example, zein protein in maize lacks in tryptophan and lysine. Now, if you consume poor proteins for a long period of time, then your body goes in negative nitrogen balance and you start losing weight. Last classification of protein is on the basis of composition of proteins. On the basis of composition of proteins, there are three types of proteins: simple proteins, conjugated proteins, and derived proteins. Simple proteins means the proteins which are made up of only from amino acids. Therefore, these proteins on hydrolysis gives only amino acids. These simple proteins are further subclassified on the basis of solubility and coagulation as albumin, globulin, protamine, prolamine, gluteins, scleroproteins, etc. First, about albumin. It is soluble in water, coagulated by heat, precipitated by full saturation. For full saturation, we use solid ammonium sulfate powder. Its isoelectric point or isoelectric pH is 4.7. Examples of this albumin are lactalbumin present in milk, human albumin present in our blood, OA albumin in egg, leucosin is albumin present in cereal, and legumelin is a type of albumin present in legumes. Next simple protein is globulin. It is insoluble in water but soluble in dilute salt solution. It is coagulated by heat. It is precipitated by half saturation test. For half saturation test, we will use liquid ammonium sulfate solution. Examples of this are lactglobulin of milk, OA globulin of egg, legumin of legumes, globin chains of hemoglobin, immunoglobulins are the examples of these globulins. Next simple protein is protamines, which is soluble in water, dilute acid and alkali but not coagulated by heat. They are deficient in cysteine, tryptophan, tyrosine, but they are rich in arginine and lysine, which are basic amino acid. Hence, they are called as basic proteins. Examples of these protamines are salmon and sardine protein of fish is the natural source. Apart from this protamine is also associated with zinc insulinate commercial preparation, which maintains the stability of insulin. Next simple protein is prolamine, which is insoluble in water but soluble in 70 to 80% alcohol. It is deficient in lysine but rich in proline. Examples: gliadin protein of wheat and hordin protein of barley. Next simple protein is gluteins. It is insoluble in water but soluble in dilute water or alkalis. It is coagulated by heat, rich in glutamic acid. Examples of gluteins are oraisin protein of rice and glutenin protein of wheat. Now about wheat proteins. Wheat contains 4 types of proteins. They are classified into gluten proteins and non-gluten proteins. Gluten proteins means prolamine, that is gliadin, and glutenin or glutenanin. Non-gluten proteins are albumin, that is leucosin, and globulin, that is wheat globulin. Some patients have allergy for these gluten proteins. Therefore, such patients should avoid taking wheat. Next simple protein is scleroprotein. It is insoluble in water and soluble in hot and strong acids. Example: collagen, elastin, keratin. Collagen is found in connective tissue, bone, cartilage and tendons. Elastin is also found in connective tissue, ligament and tendons. Keratin is found in nails, hairs and horn hooves. After completing simple protein, we will see now conjugated proteins. Conjugated proteins are the proteins which are made up of simple proteins, that is amino acid plus other compounds. Here the proteins are called as apoproteins and other compound is called as prosthetic group, and combination of these two are called as holoproteins. Examples of conjugated protein includes glycoproteins. It contains carbohydrate along with protein. Examples of glycoproteins are blood group antigens which are present on RBCs, immunoglobulins, serum proteins. All of our serum proteins are glycoproteins, means they combines with small amount of carbohydrates. Next conjugated protein is nucleoproteins. Here the other compound or prosthetic group is nucleic acid. Histone is a protein which combines with DNA to form nucleoproteins. Next is mucoproteins or mucopolysaccharides, which are present in ground substances of connective tissue, bones, cartilage and tendons. In these mucoproteins, the mucopolysaccharides are present along with proteins. Chromoproteins are also examples of conjugated proteins where colored pigments are associated with proteins. Hemoglobin, where prosthetic group is heme. Flavoproteins, where the prosthetic group is FAD or FMN. Rhodopsin, where the prosthetic group is opsin. Prosthetic group is retinal. Next, phosphoprotein, which contains proteins and phosphate. Example, casein of milk, O-vitelline of egg. Lipoproteins, as the name suggests, it is a protein associated with lipids. Examples, chylomicron, very low density lipoprotein VLDL, low density lipoprotein LDL, high density lipoprotein HDL. And next are metalloproteins, means the proteins which are associated with metals. Ferritin is a protein which contain iron as a prosthetic group. Ceruloplasmin contain copper. Carbonic anhydrase contains zinc. So these are the examples of conjugated proteins. Now last is derived proteins. The proteins which are obtained from simple proteins and conjugated proteins by physical or chemical factors, then they are called as derived proteins. Derived proteins, again there are two types, primary derived proteins and secondary derived proteins. Primary derived proteins are formed by denaturation or coagulation of proteins. Here the primary structure of protein is intact and thus peptide bonds are also intact. No loss of amino acid in these primary derived proteins as compared to original proteins. The factors which causes denaturation or coagulation are heat, X-ray, ultraviolet rays, vigorous shaking, acid, alkali, etc. Three types of primary derived proteins are there. First category is proteins found in fibrin, myosin, elastin. Metaproteins, examples acid metaproteins, alkali metaproteins. Coagulated proteins, example cooked meat, cooked egg, albumin. So these are three types of primary derived proteins. Secondary derived proteins means these are the proteins formed by hydrolysis of peptide bond. Means some of the amino acids are lost from original proteins. There are further three types: proteases, peptones and peptides. When some amino acids are lost from proteins, they are called as proteases. When more amino acids are lost, they are called as peptones. Again, when peptones loses few amino acids, then they are called as peptides. So this is about proteins and its various classification. Thank you very much.